Greetings everyone, and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Advancing Early Cardiovascular Drug Discovery. I'm Jeff Lugaliskas, Technical Editor for Gen, and I'll be the moderator for today's webinar presentation. Through the adoption of the so-called open research model, which dispenses with recurrent fixed costs in favor of a more flexible and dynamic external spend, biopharma companies can tap into technological advances that go far beyond their own borders. This scenario allows for continued innovation, even as research budgets are coming under ever-increasing pressure. Let's meet our speakers for this webinar, who will describe how the use of an integrated approach that fuses the disciplines of in silico drug design, medicinal chemistry, and discovery biology help them meet key goals during the early discovery process. Additionally, our presenters will highlight the value of their open collaboration while focusing on discovery campaigns that employ highly relevant functional assays. Our first presentation will be given by Kenneth Werdman, Senior Vice President at Ikogen. Dr. Werdman will provide a brief introduction to Ikogen as a company and some of its current research into muscle biology and associated disorders. Our second presentation will be given by Robert McDowell, Senior Vice President of Drug Discovery at Myocardia. Dr. McDowell will tell us about cardiac muscle disruptions associated with cardiomyopathy and Myocardia's collaboration with Ikogen to assist the identification of alternative pathways affecting cardiomyopathy. Before we get rolling into Ken's presentation, I want to encourage everyone to submit questions for our Q&A session at the end of the talks. We'll try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. Okay, with all the logistics out of the way, let's get our webinar started. Ken, the audience is listening.
Thank you, Jeff. Hello, my name is Ken Werdman, and I'm the Director of the Ikogen Research Center in Tucson, Arizona. I have two major objectives today. The first is to introduce you to our company, Ikogen, and the second, I'd like to share with you a bit of our exploratory studies in muscle biology and rare disease. I'm pleased to be sharing this venue with our long-term collaborator, Dr. Robert McDowell from the company Myocardia in South San Francisco. Bob's presentation will share a story of collaboration between our teams in the context of Myocardia's cardiovascular research. So first, who is Ikogen? Ikogen is a partnership research organization, or a PRO, specializing in the early phase of drug discovery research. Our efforts are driven by strong capabilities in high-performance computing, ultra-high throughput screening, and automated chemistry. Our interest is to provide turnkey solutions for the drug discovery needs of partners in all sectors from big pharma to virtual startups. Underpinning these technical capabilities is a deeply experienced workforce. It's a team with an average tenure in excess of 15 years. Many of our folks have experience gleaned from dozens of discovery programs and have delivered numerous lead compounds into legacy company projects, among which a handful of these compounds have successfully progressed to clinical phases. This team has honed its craft working within major pharma through collaboration with therapeutic partners. The focus of this team has always been on the early phases from therapeutic concept to candidate. Thus, we have complete knowledge in-house to plan, build, and execute early drug discovery research programs. At this point, some of you may be thinking, I know Ikogen, they're an ion channel research company in North Carolina. This statement was correct up until July of 2016, when Ikogen acquired an integrated drug discovery site from Sanofi in Arizona. With this acquisition, Ikogen transformed from a world-class ion channel company into a fully capable drug discovery engine, acquiring ultra-high throughput capabilities in chemistry, biology, and computation, and the experienced workforce described in my previous slide. Together, these two teams offer big pharma capabilities and the experience to deliver on drug discovery objectives from partners across North America. In terms of capabilities, Ikogen biology is fully enabled with a broad range of assay types and detection technologies. This is complemented by deep expertise in cell biology, including the manipulation of primary cells and stem cells, having executed past discovery work employing, among other types, neurons, oligodendrocytes, chondrocytes, muscle cells, and their progenitors. The team is empowered by state-of-the-art robotics and a suite of capabilities which supports true ultra-high throughput screening and active sample follow-up. Biology's capabilities include the manipulation and analysis of cellular systems by a variety of methods, including high-content imaging and high-throughput transcriptional response profile. And finally, we have in-house ADME screening to support projects with rapid assessment of the pharmaceutical properties of advancing chemical series. Chemists at Erika Jen have two key mandates. The first is to drive the medicinal optimization of active chemical series discovered through screening, which is an area within which they possess significant experience. Second, through a suite of automated chemistries, the chemists drive the evolution of Erika Jen's novel screening collection. This work includes elaborating novel sets based on recently published discoveries, expanding series of interesting chemotypes found during screening, and the creation of special purpose compound collections. This activity is supported by analytics, purification, and sample management automation. Overall, chemistry operates at a throughput scale to fully leverage the high throughput capabilities of the site. Also of note is an exceptional knowledge in peptide chemistry, which we can employ for the optimization of peptides or similar compounds, macrocyclic libraries, or on the larger side, even grafting functional epitopes into mini proteins. Recently, we have made significant investments in the field of high-performance computing and computational chemistry, developing methods that accelerate and enlighten teams throughout project progression. Our success in predicting the properties of molecules prior to synthesis, both for libraries and optimization arrays, has extended to the prediction of bioactivity based on target or ligand structure, what some call virtual screening. Most recently, we're using force-field methods to perform long time-scale molecular dynamic simulations in order to incorporate target and ligand flexibility to increase the accuracy of these predictive models. Indeed, today, in well-controlled systems, we can predict the relative affinity of ligands for their target prior to synthesis. This fundamentally changes the game for multi-parametric optimization of lead compounds. But as powerful as these individual technologies are, magic really begins when they are wielded by an interdisciplinary team with deep expertise, strong interpersonal relationships, and a clear mission. One of my favorite quotes is that of Dwight D. Eisenhower, who said, "In preparing for battle, I have always found that plans are useless, but planning is indispensable." Quality research, like battle, is a process of continuously assessing conditions, challenges, and opportunities, of choosing best options, not discipline by discipline, but with a view that overarches the project. We strive to achieve this through the assembly of dedicated, fully integrated project teams which interface continuously with partners to drive project progression. Over the last 2 decades of working in this fashion, we have built a conceptual framework and operational environment which is optimized for interdisciplinary drug discovery. From the building of assays needed for the creation of a well-designed program, executing informative screening campaigns, characterizing the properties of active samples, or selecting the best chemical series for therapeutic development. Throughout project progression, biological, physical, and safety data are framed in the context of public information to enlighten teams and create computational models that inform on compound properties and catalyze team thinking. However, just finding compounds with right in vitro activity and good pharmacological properties does not guarantee success if the underlying biology is uninformative or worse, perhaps irrelevant. For this reason, we have focused our efforts recently on bringing relevant human biology into the lead discovery and series selection activities. In the next few minutes, I'd like to cameo a few examples of our efforts to bring human biology into our discovery programs. And while not in the cardiovascular field per se, what I will describe is within the mesoderm lineage, in the area of human skeletal muscle development. Mature muscle requires a repair and regeneration capacity, and at the heart of this process is a quiescent progenitor cell residing between the basal lamina and the sarcolemma of the muscle fiber, called the satellite cell. When activated by exercise or other sources of muscle damage, satellite cells awaken and proliferate, creating a population of myoblasts which mature to myocytes and ultimately fuse and renew the underlying muscle cell. The satellite cell's transition through this process is fairly well characterized, and populations of differentiating cells can be readily evaluated using a handful of biomarkers. We were interested in using primary satellite cells in the discovery of therapeutics for people with Duchenne's muscular dystrophy. DMD is a genetic disorder characterized by progressive muscle degeneration and weakness, and to this end, we use satellite cell markers to purify a population of primary human satellites by fluorescence-activated cell sorting, starting with postmortem DMD tissue, and employed these isolated satellite cells to screen for compounds that would stimulate them to enter proliferation. In an automated screen of 11,000 compounds of known mechanism of action, we identified 7 that reproducibly enhanced proliferation of satellite cells into myoblasts. These experiments taught us a great deal about the isolation and handling of satellite cells and myoblasts in high-throughput processes. In many ways, however, the experiment lacks sufficient sophistication. First, it only looks at the proliferation of these cells in vitro, and does so without resolving aspects of differentiation. However, it prepared us well for what we really wanted to do. That was to use primary DMD satellite cells to identify compounds that would promote the expression of utrophin during myogenesis. Utrophin is a functional homolog of the dystrophin gene, and whose expression offers the potential to compensate for dystrophin loss in this disease. Scaling up, we executed a screen of over 1 million compounds in 1536 well format, measuring the abundance of utrophin through an HTRF method employing anti-utrophin antibodies. Over the course of about 3 months, we successfully identified 50 compounds which reproducibly elevate utrophin production in differentiating human DMD satellite cells. These molecules remain of interest to us today and are to be evaluated in advanced differentiation models, one of which I will describe for you in a moment. In parallel with this screening effort, we are also using these primary cells in studies designed to gain better experimental control over the muscle differentiation process and to optimize their use for the generation of mature myotubes in vitro. One of our scientists, Chris Penton, then an MDA-funded postdoctoral fellow in our lab, investigated the effect of cultivating isolated primary satellite cells on a variety of protein-coated substrates. His study showed that laminin 521 produced the best context for the expansion of satellite cell populations. On laminin 521, satellite cells can be expanded through multiple passages to yield a significant population of myoblasts, which retain the potential to be efficiently and predictably differentiated into myotubes. This method has significantly reduced the burden of primary cell isolation from tissues and has greatly accelerated our muscle programs for both human and mouse models from primary cells. For those interested in the details on these experiments, I direct you to a publication of this work last year in the journal Skeletal Muscle. Of course, these days, a common route to specific populations of human cells is through induced pluripotency, and although not speedy, the generation of iPS cells from patient populations does offer significant advantages, perhaps most important of which is the ability, through genome editing, to engineer isogenic comparator cells for experimental models or disease reporter constructs for use in high-throughput screens. We have found that the methods we developed for primary cell differentiation perform equally well for the differentiation of iPSCs into myotubes. In our standard 25-day protocol, Cells at day 8 demonstrate the morphology and properties of myocytes, and by day 19, 6 days post-shift to differentiation conditions, they begin to fuse, forming multinucleate syncytia and ultimately elaborating myotubular structures. Changes are accompanied by the loss of satellite cell marker PAX7, and over the course of differentiation show the expression of key myogenic program factors and ultimately the high-level expression of mature muscle proteins, including myosin heavy chain, troponin type 1, and desmin, the latter suggesting that some degree of sarcomeric architecture development is occurring in these models. In collaboration with Kip Parker's lab at Harvard University, we have begun to use iPSC-derived muscle cells to develop functional skeletal muscle models by growing engineered tissues as thin films, using methods analogous to those developed in the Parker lab for cardiomyocyte. In the Parker method, a template created by microfabrication is used to cast a gelatin substrate upon which myogenic progenitors can be cultured and differentiated in the muscle tissue. Over time, healthy myocytes, under influence of the patterning, create beautiful, highly ordered anisotropic tissues. In this model, normal and DMD iPSC-derived tissues demonstrate striking differences, with the DMD cells failing to develop well-ordered intercellular organization and demonstrating significant reduction in several properties of mature muscle. These morphological differences correlate with a significant change in the mechanical properties of the tissue. In this model, contraction of the oriented tissue results in a curling of the film upon which the tissue is ground. The amount of force created by the tissue can be determined by measuring the extent of deflection of the film. Here we see that both baseline and peak stress forces are reduced in tissues formed from DMD cells relative to normal cells. Our intent is to use models such as these to assess the ability of compounds found in screening to influence the development of DMD-derived cells to yield tissues with more normal functional properties. Overall, we expect that the effort taken to bring functional human endpoints into the series selection and optimization process will significantly improve the efficacy of early discovery and promises a greater likelihood of success in proof-of-concept studies. So with that, I need to conclude. I hope I've given you a sense of the new Ikogen, a team with deep experience in discovery and broad-ranging capabilities, where automation enables a speed and scale which is comparable to the best industrial team, and a team focused on changing the drug discovery game, not simply in terms of efficiency, but with respect to efficacy as well. I thank you for your time and interest, and with that, I'll pass it back to you, Joe.
Excellent. Thanks very much, Ken. That was a really great insight into Ikogen and its capabilities in the area of muscle cell research. Before we begin Bob's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the talks. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. We'll try to get to as many questions as we can. All right, let's continue with the presentations. Bob, the floor is yours.
It's a pleasure to join you all today. My name is Robert McDowell, and I head the drug discovery functions here at MyoKardia. We first started working with Ikogen as part of an ongoing collaboration with Sanofi when the Tucson group was at that time aligned with Sanofi. The work that I'm going to discuss today has launched multiple programs that are still active in our portfolio, so I'm not going to be able to share a lot of data from the programs, but instead we'll focus on the process and the impact it's had on MyoKardia as a growing company. MyoKardia was founded in 2012 by Third Rock Ventures in collaboration with a number of well-regarded academic partners with deep expertise in the cardiovascular muscle biology, this including Jim Spudich at Stanford, Leslie Leinwand at University of Colorado, and John and Cricket Seidman at Harvard University. And together, they realized that the time was right to build a company focused on developing small molecule therapeutics for heritable cardiomyopathies. The feeling was at the time that the confluence of genetic information, understanding of the underlying biophysics of the heart, and how mutations in heart proteins lead to disease phenotype provided an ideal platform with which to look for therapeutics for these unmet patient needs. Our initial focus has been on heritable cardiomyopathies, particularly subgroups of HCM and DCM, which I'll describe next. The two most common forms of heritable cardiomyopathies are hypertrophic cardiomyopathy, or HCM, and dilated cardiomyopathy, or DCM. These are a group of genetically driven diseases where mutations in the heart muscle proteins cause abnormal contraction or relaxation, leading to progressive heart failure. These are lifelong diseases for which there is no known treatment right now, short of surgery or transplantation. In HCM, the heart contracts too strongly and becomes thick and inelastic due to this excess contraction, and concomitantly then suffers from impaired relaxation, leading ultimately to ischemia. We hear from time to time about young athletes dying on the field with enlarged hearts. These are typically cases of undiagnosed HCM. In DCM, quite the opposite. The heart is thin and weakened and is unable to pump blood adequately. In both cases, either because of the excess thickening and inelasticity or the weakened strength of the heart is not able to deliver sufficient blood to peripheral tissues to avoid symptoms of heart failure. Many of the mutations that lead to HCM or DCM are found in the proteins of the contraction machinery of the heart, or the sarcomere. If we look at the heart as an organ, it's composed of a series of muscle fibers aligned in a striated array. Each muscle fiber in turn consists of multiple myofibrils, again, all bundled together in an array. And the essential contraction unit of the heart is the so-called sarcomere, which contains the thin and thick filaments where the myosin motor protein, which is tied to the thick filament, reaches up, binds onto the thin filament, and pulls it forward in a sliding motion. The so-called sliding cross-bridge hypothesis is true to all striated muscle, and particularly the heart. Our initial targets were myosin, the myosin motor itself, which contains many different mutations associated with both HCM and DCM. This is the protein that really drives contraction. So our initial programs were designed to correct these underlying causal defects in HCM. In HCM, mutations in myosin or other proteins in the sarcomere lead to too many cross bridges being formed. The net result is the heart pulls too strongly during contraction and is unable to relax adequately during relaxation because of the excess number of cross bridges attached. MYK-461, which is now in phase 2, is designed to reduce the number of cross bridges in the HCM heart and restore normal contractility. In DCM, quite the opposite. There are too few cross bridges, or the cross bridges themselves do not exert enough force. The net result in either case is inadequate contraction, and therefore a heart that does not pump strongly enough. MYK-491, which is now in phase 1, is designed to enhance the number of cross bridges and once again restore contractility. And actually some of the work that we did with the Erika Jen group led to 491, although I'm not going to describe that work in detail today. Now, the heart is a very complex, highly regulated machine, and in addition to myosin, there are many other proteins that work in concert to govern both contraction and relaxation. And as we look one layer deeper into the genetics of disease, it becomes clear that many of the mutations that drive disease result in abnormal functioning of these more subtle regulatory elements. So we wanted to tap into this as we thought about building a portfolio of compounds that would allow a physician to precisely tune a patient's heart back to normal conditions by having molecules that would address not only myosin itself, but the regulating proteins that govern how myosin functions. Our goal at MyoKardia ultimately is to generate therapies that precisely correct given defects. What I'd like to describe next is the context of the collaboration, where we were when we first started, and then shift into some of the tools that we brought to bear along with the Erika Jen team, and then describe a little bit of the outcome of the collaboration itself. So when we started working together in the fourth quarter of 2014, MyoKardia was approximately a year and a half old. At that point, we'd established our basic biochemical and cell-based platform. We had a lead preclinical candidate and backup series for HCM, and that preclinical candidate is now in phase 2. We had some early screening hits for DCM, and that program ultimately gave rise to 491, which, as I mentioned earlier, had a lot of input from the IPSC team. Our goals of the collaboration were really to identify non-myosin mechanisms for both activation and inhibition, in other words, to start to tap into some of these more subtle regulatory elements so that we could complement what we were doing with the myosin modulators, but to also generate a backup series for the activators. From its inception, one of the underlying philosophies of MyoKardia was to build a translational vision where we could gain clinical feedback from our initial programs to inform second and third generation approaches. We're really undertaking brand new therapies in an area that has not been mined before in the industry, and so we stand to learn a lot from our patients as we treat them with the first generation molecules, those being the myosin modulators. So in order to fulfill this translational vision, we really needed to get beyond myosin itself and look throughout the rest of the sarcomere. And this is really where the collaboration with IPSC has helped propel us forward. So at the outset, we wanted to maintain or establish a completely transparent collaboration where we would share resources and share idea making with the team in Tucson. In order to do this, we took a number of the assay systems that we had built at MyoKardia and replicated them in the Tucson group. And as Ken mentioned, that group already had some forward momentum in muscle biology systems. We had a number of biochemical assays that we transferred, all based on looking at the rate of phosphate release from myosin as a function of its hydrolysis of ATP. And these assays start from the very simple study of myosin itself to actomyosin, where we put myosin in the presence of actin, to the regulated thin filament, where we can reconstitute elements of the sarcomere by adding in some of the other regulatory proteins, and then finally to skin myofibrils, which represent a complete intact sarcomere isolated from natural sources. All of these involved a steady-state ATPase assay that really required a coupled assay system and was quite complex to set up, not only for routine SAR purposes, but particularly for screening. By having this full suite of biochemical assays, we could then screen phenotypically in a system like the skin myofibrils, and then use these more subtractive methods to understand which targets were required in the sarcomere were required for the activity of the compounds we identified. Those compounds were then subject to secondary studies, biophysical techniques, run here at MyoKardia. Once we were convinced that we had the right biochemistry, then we wanted to look at the impact of that biochemistry on contraction itself. For this, our workhorse tools were the cardiomyocytes, again, primary cells isolated from adult tissue, and skin muscle fibers, where we could actually look under load at not only the net force production, tension production, but also calcium sensitivity. Many of these assays, as I said, were shared in common with the group at IPSC. The myosin, actomyosin, and skin myofibril assays were installed in both sites, and IPSC, as you see, has had quite an investment in cardiomyocyte methods for some time. We also had parallel methods developed in vivo for both image-based and invasive methods to understand what the compounds were doing at the contraction level as we moved into larger systems, including live animals. But I'm not going to describe those today. So now I'd like to really highlight some of the collaboration timelines and the key success factors that I think led to this very productive relationship. I'll really touch on three major areas of work where we had overlapping work with the IPSC team itself. First and foremost is clearly the biochemistry and high-throughput screening. We screened over 1.2 million compounds in this collaboration, and this involved not only transferring the assays to IPSC, but making sure they were stable under high-throughput conditions, running the screen, and all the confirmatory assays to make sure that the hits were real. This took advantage of the full suite of biochemical methods that Ken outlined earlier in his presentation. And this started really in 2014 at the end, fourth quarter, and carried on through mid-2016. As the hits emerged, we then tapped into the computational team at Jen to help us deconvolute the hits, first of all by pharmacophore, and look for similar compounds, whether in the Jen, at that time, Sanofi collection, or available commercially, so that we could really build an early structure-activity relationship around these mechanisms while we were in the business of dissecting them out biochemically internally. And then as we got the hits confirmed and we started to see trends that we liked, particularly identifying molecules that were not working directly at myosin itself, we engaged with the chemistry team to do iterative hit-to-lead work as well, supported by all of the in vitro ADME prediction and other capabilities Ken mentioned. Our goal was to get compounds that were of sufficient quality that we could rapidly go into cells, fibers, and ultimately in vivo. In order to do this, we needed to make sure that they had good physical properties, that they were stable, and they would be devoid of off-target artifacts that could confound our interpretation of the pharmacology. So the net result of all of this was that we screened over 1.2 million compounds. We had the hits classified by mechanism by the end of the third quarter of 2015 using the subtractive method that I described earlier. With the help of the computational group, we selected both analogs and isostairs, which gave us rapid validation of structure-activity trends. And by mid-2016, we actually had identified our backup activator series with in vivo data, and that series continues today. Also, by the end of 2016, more excitingly for me, we had identified non-myosin modulators, one of which we've actually taken into in vivo studies, the others of which are poised for in vivo work later on this year. All of these are novel mechanisms that had not been described in the literature and tap into these exact regulatory elements that I described earlier. So the net result was that this collaboration produced 3 active programs, all of which are still ongoing chemistry at MyoKardia, and we anticipate generating clinical candidates out of at least 2 of them. So if I look at the collaboration from this standpoint, our key goals were clearly met. The backup activator series was identified, non-myosin modulators were identified with in vivo activity, and this in vivo activity is allowing us to now validate these mechanisms in the context of disease models that we've been building here at MyoKardia. Some of the critical success factors I think that led to this working as well as it did, this was a true collaboration on all aspects, from assay refinement, screening strategy, and hit triage. The hit-to-lead chemistry effort, again, was completely integrated between the 2 sites. There was parallel work going on at both sites, transparent sharing of all data. We set up an electronic database to exchange structure-activity data in real time, and true joint decision-making by the project teams. At any one week, there would be Jen scientists here at MyoKardia or MyoKardia scientists in the Tucson site working together to solve these problems. And it was only through that intense collaborative spirit that we got to where we did in such short time. So some concluding comments. By all accounts, this collaboration was a real success. We clearly met our discovery goals. We've identified new mechanisms in chemical matter that are now being validated in vivo, and we've actually initiated a follow-on screen in late 2016 based on a new assay concept, the screen now being conducted with Jen directly, no longer part of Sanofi. Keys to that success, as I've mentioned before, was that it was a true partnership. Shared idea generation and decision-making at both sites, close communication and collaboration across all functions, and a selfless integration that took advantage of everybody's skills. If I step back and look at MyoKardia today, the impact of the collaboration is without question. We currently have our lead program, 461, in phase 2 and 491 in phase 1. As we're learning from patients in the phase 2 program, we're gaining new insights as to what we want out of second-generation mechanisms, and the collaboration has now given us a palette of molecules and mechanisms with which to test those insights very quickly. In other words, fulfilling the translational cycle that we talked about and hoped for when the company was founded. We are anticipating new collaborations with this team as we continue to build our understanding of how the regulatory interactions in the sarcomere drive contraction, both in the healthy heart and in disease, and look forward to more success in the future.
Thanks very much, Bob. That was some really great information about MyoKardia's work into cardiomyopathy research and the benefit of its open collaboration with Ikogen. So we thank you for that. Before we start the Q&A session, I want to let everyone know this is their final chance to submit questions for our speakers. So hurry up and send them in now. All right, it looks like we've got some really great questions that have come in, so let's begin the Q&A session. Give us a few moments to get all the technological things worked out on our side, and we'll be presently. All right, everyone, let's begin the Q&A. First question is going to be for Ken. Ken, one of our audience members would like to know, what is a partnership research organization, and how does it differ from a CRO?
Thanks, Jeff. It's actually a good question. It's in many ways more than just spin and terminology. I think what Bob described is at the heart of what a partnership research organization is. It's the ability to integrate into the work with our partners, and to add value to the program, not just through technical expertise and capabilities, but by making intellectual contributions. This really requires that we have clear and transparent communication as described, and it really gives us the opportunity by the integration of activities here in Tucson to accelerate the partners program while providing them with continuous input and ability to contribute and steer. In many ways, a PRO gives you the opportunity to drive the partner's car with all the benefit of the expert navigation system, but with hands on the steering wheel. This is somehow different than a contract research organization where you really come for a discrete task and you choose your partner based on a specific niche area of expertise. There's roles for both. We actually do both, but we think we add the greatest value to our clients' programs when we can join their team and provide our expertise in a fully integrated fashion.
All right, thank you, Ken. Next question is for Bob. Bob, one of our audience members would like to know, since connexin proteins and gap junctional intercellular communication is critical for heart function, has this family of connexin genes and the GJIC been studied as targets for your drug discovery?
Yeah, thank you very much. Right now it's on our short list once we get past the contraction machinery. Now that we have molecules in phase 2 and we can look at responses in both patients but also in large animal models of disease, we want to see how much does correcting the underlying contraction defect address some of the other pathologies of disease, and if it does not, and connexin gap junction is one area, metabolism or energy usage is another, then we'll think about additional pathways that we might need to modulate. The ultimate goal, I think, will be to develop a cocktail of therapeutics that we can tune for each patient's pathology and disease progression.
Great, thank you, Bob. Next question for Ken. Ken, what is the composition of the population of cells expanded on the laminin 521? To what extent does it contain types of cells not in the muscle lineage?
Well, our best evaluation of these cultures show that they're over 90% muscle lineage cells and predominantly in a myoblast-like state.
All righty, Ken, thank you. Next question is going to be for you as well. Can these cells be frozen and be used directly?
Yeah, we will, after expanding into this myoblast population, we will freeze the cells back and can bring them directly out into experimental protocols, including differentiation to myocytes and myotubes.
All righty. Ken, we have 2 more questions for you. Next one is, how many cells can you derive by a 521 expansion, and how many passages can be made?
Well, we've taken them out on up to, I believe, unless I haven't talked to the team recently on this, but the last tally was we've been 8 expansion passages with efficient differentiation.
Alrighty, thank you. And our last question, would like to know internal ion channel inhibitors. To what extent can you rely on results of molecular modeling, particularly for compounds with lower affinity? Are experimental approaches necessary, and in what phase?
Yeah, that's a really good question in that as we bring the two teams together. As mentioned, the group in North Carolina was a former Pfizer group with specialization in ion channel biology and pharmacology. We're really interested in bringing our computational capability in Tucson and leveraging it with that expertise in North Carolina. I think there's a real chance for us to do some amazing things in the ion channel space. But that said, structural biology on ion channels is lagging behind. That was, I would say, all membrane protein structural biology. Some recent, let's say in the last, I guess, 10 years, I'm dating myself, recent, work on GPCRs is provided to bring good structural information into the drug discovery space for G protein coupled receptors. There are some innovations today in the structural biology space which will begin, I believe, to bring us true structural data for ion channels. But that's not where we are today. Today we have cryo-EM that gives us some insight into subdomain structure, and with good modeling, you can come to a point where you have some sense of the structural organization of the ion channel and start to use that to identify where pockets may be available for the purpose of developing, say, allosteric modulators of the channel activity. But this is pretty much black art at this point in time. Once you have ligands, however, the game has changed in that you start to understand how existing ligands interact with your models and refine those models to fit the FAR understanding of the compounds and begin to derive cause and effect relationships for binding interactions. And at that point, I think molecular modeling can become quite powerful in developing ion channel modulators. I would stay tuned to the structural biology on ion channels because I think it's a landscape that will be changing over the next 3 to 5 years.
Alrighty, thank you, Ken. And with that, it looks like we've come to the end of our webinar. So I want to remind everyone that the webinar will be archived on our site for up to a year at www.genengnews.com. So if you missed any parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Ken and Bob again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Erika Jen for sponsoring this webinar. So hopefully we'll see you again in the future at another Gen webinar. Goodbye for now.